Hello, everyone. The internal assessment. Dun, dun, dun. Everybody's worst enemy, but it should be your best friend. You know why? Because the highest score you can get is a 24, and that counts for 20% of your total grade. So if you take paper 1, paper 2, paper 3, this is, I'm going to make up numbers right now. I think this is 20, this is 40, and paper 3 is also 20. I think that's about right. So a fifth, pretty good math, a fifth of your total IA score is your IA. And everybody should get 7s on this, to be honest. A 7 last year was, I think, a 20 or higher. So 20 to 24 was a 7. I am here to help. So use me, drive me crazy, Zoom meetings, drop by and visit, whatever it takes, because we should all get high scores on this. So as you can see here, your internal assessment is divided into 5 categories: personal engagement, exploration, analysis, evaluation, communication. These 3 down here, we're not even going to look at till much later, because you can't even think about these until you design an experiment and collect the data. Like you have to design the experiment first, right? Also, personal engagement, I'm not even going to think about that one either. Actually, I'm not even going to ever talk about it, because the personal engagement is actually embedded in your exploration. There's a part in exploration called background, and if you write a decent background, it shows that you're engaged, that you're interested in the topic, and that's all this is really doing, is a way to assess how engaged you are. Okay, so let's start with exploration, because that's what I said. So exploration has 4 little subtopics here. The research question, important. The background, oh, you were just talking about that. So that would be the personal engagement piece. The method is where I like to start, because this is where you start thinking about how to answer your research question. And then every IA will have safety, ethics, and environmental issues addressed. Sometimes this is tough, but we can do it together. Okay, so I want to talk about the method, and in particular, what is an independent variable and what is a dependent variable. There's 3 different variables that you need to talk about. That's actually kind of a lie. There's really 4. We're going to talk about confounding variables also. But the independent variable, the dependent variable, and the controlled variables. Controlled variables we're going to do in a different video. So I'm going to try to find a cleaner piece of paper. So I'm just going to write down here. So the independent variable is what you are manipulating. It's like, you know what? I'm going to set up an experiment and I am going to set these different units to see the effect of that on something. So you control this. You design an experiment to set the independent variables. So let's pretend that you want to know how light affects the rate of photosynthesis. That's a good one. So you're going to set up an experiment and you're going to try different intensities of light. There's different ways of doing this. We have light sense meters that measure how strong the light is in lux. You could also set the light source at different distances from the plant. So let's do it that way. Let's say we did an experiment and we had distances from 10 centimeters all the way up to 50. Now, which of these values will have stronger light? Which of these will have the strongest light intensity? Did you say 10 centimeters? Then you're right? If you're closer, the light should be stronger. So actually, we have to put our numbers in reverse order here if we're going from weakest to strongest. So there's 50, 40, 30, 20, and 10. I'm being lazy and not writing them all out. So yeah, cool. We we have our increments of our independent variable that you controlled because you set up the different distances. The dependent variable, oh, look at that, Mr. Fleming, you're drawing a graph. The dependent variable is how you will measure. How could we measure photosynthesis? We can measure growth. Oh my goodness, that would take forever. But yeah, the plants will grow at different rates depending on how much photosynthesis is happening. Oh wait, plants need water. Tough to measure in the soil. Ooh, they use carbon dioxide. Yeah, you could measure the amount of carbon dioxide used, right? Because carbon dioxide is the ingredient of photosynthesis. So carbon dioxide consumed. You could also, if you wanted, oxygen produced, because that is a waste product. I recommend using waste products when you can, but it doesn't matter. It doesn't matter, right? So I'm going to go with the oxygen one because it's my video. So here's my y-axis, and then we do our experiment. And we'll talk about how many measurements you need at each one of these. But, you know, at 50, the oxygen produced was like this much. Maybe it was like this much here. We had a high number here, then a weird low one here, and then something like that. So you got like some sort of graph like that. So independent variables are always going to be on your x-axis when you're organizing them, and dependent variables will always, whoops, sorry, just use the eraser, Mr. Fleming. Dependent variables will always be on the y-axis. So that's independent dependent variables. And the next video, we'll talk a little bit about controlled variables and also the research question.